days, Monday, Wednesday, Friday, on the same chapter, chapter 35, you must have to wonder, man, this must be a really important chapter. And it is, because it integrates all sorts of stuff from the fall. And we're going to talk about homeostasis, we're going to talk about cell signaling, we're going to talk about phagocytosis, we're going to talk about all sorts of different things. And so we're going to take this a little bit at a time. The vocabulary in this unit, or in this chapter in particular, is extensive. So do yourself a favor and do not wait to get behind the vocabulary. Make your flashcards or whatnot. No matter what class you took for Honors Bio, you have not done any of this. This is new for everyone. So make sure that you're ready for that. Okay, and you've got your, good biology student have on from the very beginning. This is a macrophage. It's a kind of white blood cell. Okay, and these are bacteria. Macrophage is big eater. Macro, big, phage, eater. Sometimes I'm a big eater. So look at it, floating around in the body, finding things that shouldn't be there, and eating them. Well, how do they eat them? They eat them by phagocytosis. And that was a process that we first learned about back in Unit 2. So a pathogen is a thing that makes you sick. Okay, a pathogen could be a bacteria, could be a virus, could be a fungus, could be a parasite, right? Any of these things. A pathogen is something that makes you sick. For pathogens, the internal environment of an animal is an ideal habitat. There's food, water, shelter, space, means of transportation. All of these things are true. Okay, and so for them, for these viruses and bacteria, you are a perfect habitat. That's not ideal. That's not what I'd like, but I don't have a lot of control over that. So together, all of your defenses make up the immune system. The immune system enables an animal to avoid or limit many infections. And you might be like, well, that's kind of not true, as Chris coughs, right? Because we get sick. Sometimes, some of us more than others, we get sick, right? So clearly the system has its flaws. But we're going to go through and we're going to talk about that. So your first line of defense for the immune system makes an awful lot of sense. We want to keep pathogens out. Goal 1, keep them out. This is what we're looking to do. But we cannot seal off our body surfaces completely because we have to do things like breathe. And eat. Anybody want to munch on something? I mean, you're like on point. I like it. I'm going to drink, that'll work. Okay, so we have to do things like eat and drink, right? And breathe. And so we have to let stuff in order to survive. The trick is we have to let the good stuff in, but not the bad stuff in. And they can be kind of tricky to tell the two apart. Fair enough? So when stuff comes in, and it shouldn't be there, non-self. So the stuff that should be there is self, me. I should be, my cells should all be me. We call that self. Non-self invaders shouldn't be there. When they get in, we have to identify them and destroy them. It is full-on warfare inside your body, especially Chris's. Active warfare. Okay? How do we do that? How do we tell self from non-self? We do that by molecular recognition. Okay? We do that by shape. Remember when we talked about molecular shape? Conformation of molecules meant everything. Okay? That's how we accomplish this. All animals have some kind of innate immunity. Innate immunity is very fast, but it's broad and it's not specific to what's made you sick. Here's an example. You guys may have noticed, may not have noticed, just the sheer number of dandelions that grow on our lawn out here in the spring. I mean, I don't know that there's as much grass as there is dandelions out here in the Grandview High School lawn. But that's okay. It's green, and it reminds me of spring, so we'll take it. Let's suggest I wanted to kill the dandelions. There are two ways I could go about this. Method 1, I go to Home Depot, I get Roundup Industrial Strength, Gigantor backpack-sized spray bottle, dump it all in there, and I walk around and I just spray. Am I going to kill all those weeds? Am I going to kill all the lawn with it? Absolutely I am. Mission accomplished? Yes. The dandelions are all gone. It was fast, but it was non-specific. I killed a lot of things besides dandelions when I did that. In contrast to that, if I put on a pair of my leather gardening gloves, busted out a nice large hat, nobody needs a burned nose, and plucked them one at a time, could I accomplish the same goal?
Yes.
Yes. Would it take longer?
Yes.
An awful lot longer. So my analogy, my dandelion analogy, is the difference between innate immunity, which is fast but non-specific, where everything dies, versus what we call adaptive immunity. Adaptive immunity is very slow, but it's very specific, where the only things that die are the things that make you sick. Okay? The adaptive immunity learns. Okay? It learns the things that are in your environment that make you sick, so that you can only get sick by one thing one time. Adaptive immunity. Okay? So we heard about this, you may have heard about this way back in the fall, when Ebola was just wrapping up its really rough attack in many parts of Africa. Many aid workers from North America had gone to Africa to support the goal of saving lives, but some of them actually became ill while they were there with Ebola. The first guy, that once you become sick with Ebola, if you're an aid worker over there, they immediately bring you back to the States because there's a much better chance that we're going to be able to save you with our technology. So they brought back the first guy that got sick, and he lived, which it was touch and go for a while. They weren't sure if he was going to make it, but he lived. And then subsequently, two other health workers became sick and came back as well. And if you followed the story at all, they actually took the first guy that lived, and they took blood plasma from him in huge quantities. Blood plasma is the liquid part of your blood, not your red blood cells. Okay. And his blood plasma had the secret, quote unquote, or they were hoping his blood plasma contained the secret to getting rid of the Ebola in these other people. So they took his blood plasma and injected it into these other people because his immune system knew how to beat Ebola. And you've probably heard somebody say you can't catch the same cold twice. Well, you can't catch the same Ebola twice either. And so what the goal was is that this guy's immune system, the chemicals that were left behind, would stimulate these other individuals' immune systems to also beat Ebola. And it worked. And it worked. And the fact that it worked began this process of trying to come up with an immunization. Because what an immunization actually is tricking your immune system into thinking that it's already been sick with that, because you can't get the same sickness twice. So that's what this is saying. Your adaptive immunity can learn to identify pathogens more quickly the second time you encounter them. Sometimes we try to trick your immune system into thinking it's already seen it once. And that's what injections do. That's what vaccines do. Okay. Here's a diagram. You're not going to have time to fill this all in. Okay, but you could get just the basics here. Innate immunity is that top box. Adaptive immunity is that bottom box. Pathogens encounter that innate immunity first. Well, that makes sense, because the first thing you're going to try to do is keep it out. That's part of your innate or inborn defense. And then after that, you have some innate internal defenses also. This is the roundup part, right? The part of your immunity where your cells are just killing things that don't look right, but we're not really sure why. Okay? Adaptive immunity is present only in vertebrates, whereas innate immunity is present in all animals. And this part, when vertebrates get sick, we have to clean up the humors, those are the liquids of your body, as well as the cells. Two different ways to go about achieving that. This portion, this right up here, the yellow box, is today's lecture. The blue box down here is the lecture for next 2 classes. So today we're going to get through these topics here. This is where life gets much more complicated, so we'll spend 2 full days doing the bottom blue box. But it makes sense that the pathogens would encounter these things in the order they're listed. First, the pathogens are going to encounter skin and mucus. If it gets past your skin and mucus, then your internal defenses, the broad but fast defenses, will come into play. And if the pathogen gets past those, then that's when we'll move on to our adaptive immunity. All of that was my preamble to the beginning of lecture. You guys remember when we learned about phagocytosis in Unit 2? Oh yeah, remember that was cellular eating. Like, okay. Remember how hard it seemed back then? It's not as hard anymore, right? These were our pseudopodia that went out and snatched something from the extracellular space. Okay. When we talked about phagocytosis, we created a vacuole, and then we attached something to that vacuole, and we called that our lysosome. Oh yeah, remember that? Our lysosomes contained digestive enzymes, and when the lysosomes attached to the vesicle, in this case the vacuole, it would dump those lysosomes into the vesicle. The lysosomes would digest whatever was brought in, breaking it down into the individual monomers. And then when we talked about it in Unit 2, we said now the cell can make use of those monomers to build whatever kind of macromolecules it needs. In this case, when we're looking at immune cells, typically the cell is ingesting so many things in such a short period of time that it's not going to hang on to the monomers. It's just going to dump them out. It doesn't need them. It's not doing this, the phagocytosis, because it needs food. It's doing this because you're sick. Okay? So then we also have exocytosis as well. I'm going to hit escape and see if I can get this to open up properly. My bar across the bottom is being annoying. Let's try this. Sorry. I guess we're just going to have a bar. This shouldn't be here. Okay, so innate defenses, all animals have them, and they are, this is what I was just talking about on that diagram earlier, we have barrier defenses that block the entry of pathogens. We have phagocytosis, cells that engulf pathogens, and then we have antimicrobial peptides. Remember that a peptide is a non-functional protein? Remember we called them polypeptides back in Unit 1, polypeptides? Our antimicrobial peptides, what is antimicrobial soap? Soap that does what? Destroys microbes? Why don't they call it antibacterial soap? Because it probably also kills things like fungus, right? So antimicrobial is like, I'm destroying all microbes. Okay. So antimicrobial peptides are these groups of not yet functional proteins that are floating around your body looking for things that can make you sick. When they are activated, when they find things that can make you sick, they become active, and that's when they become proteins. Unique to vertebrates, there are also innate defenses called natural killer cells, NKCs. Those guys are floating around your body. I love that I have things floating around my body called NKCs. I think that's awesome. They release chemicals that cause apoptosis of an invading cell or cause apoptosis of self cells if they're sick. For example, if I have become infected with a virus and the virus is inside one of my cells, what's it doing? It's hijacked my cellular machinery, using it to duplicate lots more viruses. If a natural killer cell can find that one cell of mine that has been infected and kill it before it can produce literally millions of other viruses, is that worth it? Yeah. Does it make sense to kill the one infected cell to prevent the viruses from infecting millions of other cells? Yes. And natural killer cells do that. They will kill self cells that are not healthy, either due to viral infection or due to cancer. So that's innate. We also have things called interferons that interfere with viruses, proteins also. And then we have something called the inflammatory response. The inflammatory response is increased blood supply to an injury. If you've ever gotten a paper cut, even when you dry, even if you don't bleed, sometimes you get paper cuts that don't bleed, right? You will get red and swollen. That's your inflammatory response. That is part of your innate immune system. Barrier defenses first. Remember, the first goal of your immune system is to keep stuff out. So the barrier defenses will block the entry of pathogens to the body. So first thing, skin. A complete skin is the best thing to keep stuff out. But then, because you have to do things like eat and drink and breathe, any of those access points that you have, most of which are in your face, secrete fluid. Lots and lots of fluid. Sometimes more than others. Why? Well, because these mucous membranes that line all of this stuff that meets the outside world, if we're pushing fluids outward, it's much more difficult for pathogens to make it in. Okay? Body secretions themselves, these fluids, aside from just going against the grain, they're hostile to microbes. Lysozymes in tears, saliva destroys the cell walls of a lot of different bacteria before they can get in. Stomach fluids are highly acidic. Things you eat, the number one way for pathogens to get into your body. I think we've all probably eaten something bad at some point in our lives. You know how quickly that can come on and just destroy you. Okay? Eating is the number one way that these pathogens can make their way into your body. Because you eat all the time. We don't have a choice. We're into it. This is crazy to me. Secretions from oil and sweat glands give human skin a pH between 3 and 5. 3 and 5. I feel like things should sizzle when I touch them. 3? An acid of 3? That's like batteries, right? So how come I'm not like burning? Why doesn't the table sizzle? Quantity is so small. Okay, we talk about producing oils. I know it seems like we're oily, right? Right here for me, right here. Seems like we're oily. But in reality, the quantity of oil is so small that having a pH of 3 isn't enough to actually change things. Okay, why? Bacteria can't grow in that kind of an environment, and that's good. I don't care to be covered in bacteria. That's best for me. But there are some pathogens that make it past those barriers and make it inside. At that point, they might encounter phagocytic cells. Phagocytic, so phago is eating, cytic is cell, so that's like cell eating cell, okay? These cells detect fungal or bacterial components using membrane-bound receptors. They are called TLRs, toll-like receptors. Each TLR binds to fragments of molecules that are characteristic of pathogens. For example, is there any good reason for you to have double-stranded RNA in your body? No. All natural, healthy RNA in your body is single-stranded. But who might have double-stranded RNA? Viruses, right? So a TLR, in fact, TLR number 3, all it does is looks for double-stranded RNA. And if it finds it, it goes, warning, 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 viral, 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 and just puts everybody on alert. Similar to lipopolysaccharides. Should we have lipopolysaccharides in our body? No! That lives only on bacteria. If we find lipopolysaccharides inside of our body, something is wrong. So the cells, these phagocytic cells, have these proteins on their surface called TLRs, and all those TLRs do is look for a particular shape. And they're looking for a shape that should not be found. And if it is found, then it raises the flag. Invader alert, right? That's what we're doing. TLR5 binds to flagellin, a bacterial protein. That's not a good thing for you to have in your body. Okay. So it would raise the alarm if it were to find that. All of these macromolecules are not normally present in the body, but it is a component for groups of pathogens. Once detected by the TLR, so TLR finds it and has raised the alarm, these invading macromolecules trigger phagocytosis or destruction. Turns out you have 4 different kinds of phagocytic cells: neutrophils, macrophages, dendritic cells, and eosinophils. Chris knows all of them. Anatomy and physiology coming into play. We're going to start at the bottom with our eosinophils. Eosinophils, I think, are the most disgusting, but it's just because of what they do. They deal with multicellular invaders, like parasites. That's kind of vile to me because I'm not really a fan of worms that live inside of me. I don't think it's ever happened. I mean, maybe I'm wrong, but hopefully my eosinophils are a bored bunch of cells, right? That's the goal. Eosinophils. They discharge destructive enzymes to destroy these multicellular invaders, eosinophils. Dendritic cells are super awesome, and I would encourage you to put a star next to dendritic cells because we're going to come up with them again later in the week. Dendritic cells populate tissues that are in contact with the environment. So dendritic cells are in your skin, okay, and other areas that are in contact with the environment. Their job, in addition to phagocytosis, to consume invaders, is to then stimulate the adaptive immunity. That cell is the link between the innate immunity and the adaptive immunity. Okay, so we want to keep track of our dendritic cells. Let's put a star next to it. But then we also have neutrophils and macrophages. Neutrophils are attracted to infected tissues and engulf and destroy whatever is causing the infection, whereas macrophages migrate through the body, through the blood and the lymph, but are concentrated where pathogens are most likely to be encountered, such as areas like the spleen. All 4 of them, in general, do the same thing. They eat the stuff that make you sick. Thanks, guys. That's pretty cool. Okay. We also need to talk about our NKCs, our natural killer cells. That's what I was talking about earlier. They are looking for cell surface proteins on self cells that would be characteristic of a cell that is infected with a virus or has cancer. Natural killer cells do not engulf these cells. They release chemicals that stimulate them to undergo apoptosis, and that will just prevent further spread. So the natural killer cells aren't engulfing your sick cells. They're just stimulating them to go through apoptosis and go through programmed cell death. But once the pathogen has been recognized in the body, then we have these antimicrobial peptides that are released to limit the spread of any pathogen that may not have been neutralized in this process. 2 different kinds of antimicrobial peptides. We have interferons. I love what they do. Interferons interfere. Good job. They are proteins that interfere with viral infections. How do they do that? Well, they induce nearby healthy cells to be on the lookout. So there's one cell that got infected with a virus, and as the virus has hijacked it to cause it to make more viruses, that sick cell is releasing cues to all the ones nearby that's like, hey guys, don't let any viruses in. There's one in me. You better be careful. Be on the lookout. Right? That's what these interferons are doing. This limits that cell-to-cell spread of viruses. And then we also have a whole system called the complement system. It consists of about 30 different proteins that circulate in the plasma, in your blood. Normally in their inactive form, but they're activated when they encounter a substance that needs to be neutralized. What do they end up doing? Life-invading cells. So again, these are non-specific responses. They find your innate immunity finds a group. Like this could be a bacteria, this could be a virus, this could be a fungus, and it's attacking the group. We also have the inflammatory response, which is usually indicated by a little bit of localized pain and swelling. If you get a paper cut, the cut hurts. It doesn't hurt on your left foot, right? It's just wherever you got cut. Okay, that's what I mean by localized pain and swelling. How does that happen? Because it happens fast. If you get a paper cut, you know it, right? But you have that like redness and swelling in a matter of minutes. Right? It doesn't take long before that happens. You guys heard of antihistamines? Yeah, you take those during allergy season. It turns out allergies are an exaggerated immune response caused by too much histamine. So you take an antihistamine to counteract the histamine that's making you sneeze and whatnot. So what is a histamine? Histamine has a role in your body, a very important role in your body. What histamine does is it recruits helpers in innate defense. It's a signaling molecule that's stored in vesicles of your connective tissue. So that when you have, in this case, a splinter, this looks very aggressive. That looks like a very nasty splinter. I wouldn't like that one bit. But when this happens, the mast cells that live in your connective tissue release histamines that then recruit help. The problem is sometimes these guys get confused and release the histamines for no good reason, and that's when we have allergies. So they're not apparently a bright bunch. Okay. So what do the histamines do? Well, remember how I told you last class that your blood is always contained in vessels? But when this happens, the histamines cause these little holes in your vessels, I know they're tough to see, to widen, and for this to widen, dilate. So that your vessel becomes much more porous and there's more blood flow to that area because it's dilated. So when that happens, the temperature goes up. Look at the width. See how narrow it is here and how wide it is here? We've dilated, okay, that vessel. But then also, you can see our little pores right here. Our blood is seeping out. Okay? These are red blood cells in here. Okay? And these are going to be your neutrophils. So what happens, we got a splinter. I pulled out my tweezers and plucked it free. Now I just have to wait for it to heal. The histamines that were released here recruited help. They came over, they munched on all of my bacteria, cleaned it up. The skin is now clean, which means it will heal. That's what's happening up here. And the phagocytosis continues here. And in the end, you might have a little pus pocket. You probably all have a little pus pocket. Pus is white blood cells. I know, disgusting. Pus is a sign of your immune system doing what it's supposed to do. Pus is a sign of infection because it is your white blood cells that have attacked pathogens, and then once they have attacked the pathogens, they die. And so the reason pus is white is because they're all your white blood cells. Kind of gross. But we like that because otherwise we would just be one inflamed, disgusting mess. Severe tissue damage could lead to what we call a systemic response. It occurs throughout your entire body, where your injured cells secrete molecules that stimulate the release of extra neutrophils from the bone marrow. So if you're really sick, you have an infection, and your doctor's like, I have no idea what's going on. You seem sicker than it seems like you should be. They'll take blood, and what they're looking for is your white blood count. Okay? What they're looking to see is if you have some kind of internal systemic infection, your white blood count, white blood cell count, will be through the roof because your body's trying to fight it off. So that's what they're doing. That's what they're looking for, to figure out how severe of an infection do you actually have right now. That white blood count, white blood cell count, can increase several-fold in just a few hours. And those cells are coming from your bone marrow. The purpose of your bone marrow is to stimulate your immune system. Fever is another example of a systemic inflammatory response. Macrophages themselves can also release substances that cause the body's thermostat, remember that was your hypothalamus, to have a different set point than the 37 degrees it normally does. So through all of this, it seems foolproof. Except we still sometimes get sick. Because if you have been invaded by a virus, all of these things have been tried, and if you start to get sick, all of these things have failed. Pathogens can evade phagocytic cells and just avoid destruction completely. Remember, the viruses often have an envelope of your membrane that it can use to hide. The outer capsule and bacteria can prevent recognition. That's what happens with strep. Or bacteria themselves can resist the lysosomes even after they've been engulfed by the phagocytic cells. Tuberculosis does that. So rather than being destroyed by the host cells, tuberculosis just grows and reproduces, and the body can't really do anything about it until your adaptive immune system comes into play. All right, so you guys are going to go off to college here. Some of you in like 4 months, and some of you in a couple of years and 4 months. But nonetheless, dorm rooms are like Petri dishes. It's true. That many people living in that tiny of a space, it's like one person gets sick and it's just like you can watch it move down the hall. Part of your dorm room requirements is that you will have to have paid for the health fee or the clinic fee or whatever. And so then there becomes this question of when do you go see the doctor? Because there's going to be one on campus. That's how it works. If your throat ever looks like this, you are sick and you require medical attention. These are colonies. Remember when we did our lab with the bacteria and we grew a lawn of bacteria on our plate? That's them on your tonsils. Ugh. So it's possible that your throat could look like this and it not be strep. But if you ever look in the back of your throat and you see something that looks like a Petri dish, you should have that looked at. Okay? It's just been my experience that when kids go away to college, one of two things happens. You either go to the doctor every time something seems a little bit weird, or you never go at all. And then if you never go at all, you'll be sick forever, right? So be the smart biologist. And, you know, when you're not feeling well, you go to the bathroom with your flashlight. And if you see colonies, off you go. I got this off of the urgent care website. This is hilarious to me. Oh, was it, do I have strep throat? It's viral if things are just red and swollen. That's what they suggest. But if you have colonies, it's bacterial. That's when you go, this is what we just discussed. But I love this last symptom. Gray furry tongue. You ever find yourself with a gray furry tongue? That's another good reason to get thee to the clinic. I give you permission to skip class. Come. That's so disgusting. Okay? But that's how you can tell the difference between a virus and bacteria. Okay. And of course, if you can't sleep or you can't breathe, you know, you go to the clinic in those kinds of conditions. But sometimes it's hard to know, right? Am I really ill, or do I just have a cold and I need to tough it out? Okay. Look for the colonies. Cool.